Part 1. Genetic variation is present in most populations and species. Macro and macroevolution. Microevolution is an evolutionary change within populations of a species, and macroevolution is an evolutionary change leading to emergence of new species and other taxonomic groups. Population, it is a group of individuals belonging to the same species. They live in the same geographic area and actually can potentially interbreed. The gene pool of a population is all alleles present in a population. It is genetic information carried by members of population. The more population contain a high degree of heterozygosity. Genetic variation can be detected using artificial selection. If genetic variation does exist, phenotype will change over generations. Example, the domestic dog. Genetic and archaeological evidence indicates the domestication of dogs took place at least 15,000 years ago. Selection of desired traits present in genetic variation. Selection of desired trait present in genetic variation wild wolves. The most direct way to estimate genetic variation is to compare nucleotide sequences of genes carried by individuals in the population. For example, alcohol dehydrogenase. Alcohol dehydrogenase is a gene that determines how drunk you can be, because alcohol dehydrogenase is an enzyme that breaks down alcohol. If you have very little alcohol dehydrogenase in your liver, meaning that you can get drunk very easily because your liver cannot break down alcohol quickly in time. Compare it with Germans or any European. Any Europeans, especially the Irish and German, have a lot of alcohol dehydrogenase in their liver, in that they can actually drink a lot of beer or alcohol before getting actually drunk, in that they can drink a lot of alcohol compared to Asians. Now for ADH in Drosophila melanogaster, the gene ADH has two alleles, ADHF and ADHS. The coded protein differ by a single amino acid, histidine versus lysine at codon 1 and 2. ADH genes from natural population were cloned and sequenced. So here is the allele with all of the genes for ADH. You have exon 1, then 100 base pairs, exon 2, 3, 4. Exon 1, 2, 3, 4 are in a protein encoding region. And then you have a red area which is called the polyadenylation site, which is an untranslated region, meaning that the RNA polymerase does not touch this site. So the consensus ADH sequence: exon 3 CCCC, intron 3 GGAT, exon 4 CTC, CAC, TAG. But in the strain WAS, there are some differences. Exon 3, you have TTA instead of CCC. Intron 3, you have CATA instead of GGAAT. And exon 4, the first two codons are AC set. So FL1S also have a lot of differences. JAS have differences only in exon 4. FIF still have the same exon 3 and intron 3, but exon 4 is totally different. JAF would have a lot of differences. In exon 3, it has one codon difference. Intron 3, one codon difference. Exon 4, 6 codon difference. The ADH genes from individuals representing 5 natural populations of Drosophila were cloned, and the cloned gene contained 43 of nucleotides variations in ADH sequence of 2,721 base pairs. Out of 14 variation encoding regions, only one leads to amino acid replacement. Now in humans, CF gene. It is the one that encodes cystic fibrosis transmembrane conductance regulator, which is CFTR. Recessive loss of function mutation causes cystic fibrosis, which affects secretory glands and lungs. More than 1,900 different mutations of the CFTR gene have been identified. So these are all of the mutations that can happen towards this long sequence of genes. You have how many codons? 1, 2, 3, all the way to 24. There are 24 alleles. Right. And so the alleles between 3 and 8, it is for membrane spanning. 9 to 12, it is ATP binding. 13, 14 is R domain. And 14b to 18, you have membrane spanning. And 20 to 24, another one about ATP binding. Mean that if this is like your car can break down in many different ways. Engine can break down, the gear can break down, maybe the injection system can break down, maybe the air conditioning system can break down, maybe the belts can just snap. So all of these can result in your car unable to start, unable to move. So mean that in the CF gene, any mutation in this area can cause CF to happen. Neutral theory. Genetic variation happens due to mutation drift. So if proposal mutation leading to amino acid substitutions are detrimental, usually mutations, very serious business. You do not want any mutation to happen. So only a small fraction of mutation is favorable. So the alternative explanation for high genetic variation is natural selection. Enzyme or protein variations maintained by adaptation to certain environmental conditions. Mean that usually nature wants to stay normal unless the abnormality can cause you to live longer. Mean that nature selects you. Even though you're abnormal compared to a regular human, you are selected because you can live longer. Next section, Hardy-Weinberg Law describes allele frequencies in genotype frequencies and in populations. What is the Hardy-Weinberg Law? Hardy-Weinberg Law describes what happens to an allele or genotype frequencies in an ideal population. So what is an ideal population? An ideal population is a population which is infinitely large, with random mating, not subject to evolutionary forces such as mutation, migration, or selection. So the HW law makes two predictions. One prediction is frequency of alleles in gene pool does not change over time. Second prediction would be after one generation of random mating, genotype frequencies for two alleles is calculated as p squared plus 2pq plus q squared is 1. So p, it is the frequency of allele big A. Q is frequency of allele small a. The two allele system. It is the expected genotypic frequencies for population in Hardy-Weinberg equilibrium. It is calculated based on allele frequencies in gametes and random mating. So here, FRA, FR small a. So when you do a punnett, you can just multiply them. Let's say that the FRA, the frequency of big A is 0.7, frequency of small a is 0.3. So what is the frequency for big A, big A? We just multiply it. 0.7 multiplied by 0.7, you get 0.49. What about frequency for big A, small a? Frequency for big A, small a, 0.7 times 0.3, it is 0.21. What about small a, big A? 0.3 times 0.7, you get 0.21. What about small a, 0.3 times 0.3, 0.09. So there you have it. Frequency for big A is 0.49. Frequency for big A, small a, which is two of them, it is 2 multiplied by 0.21, so you get 0.42. And the frequency for small a is 0.09. So if we simplify it, we can say that frequency for big A is P times P squared. And then what about QP and PQ? They are both same, so we can put it as 2PQ. Frequency for small a, it is Q times Q, which is Q squared. So you can see P squared plus PQ plus QP, which is, we can simplify as 2PQ plus Q squared is the Hardy-Weinberg equation. So the Hardy-Weinberg model assumes that there will be equal rate of survival and reproduction, no selection, no new alleles arise or created by mutation, no migration into or out of population, infinitely large population, or random mating occurs. Hardy-Weinberg law has three additional consequences. The dominant traits do not necessarily increase from one generation to the next. Genetic variability can be maintained. By known frequency of one genotype, frequencies of other genotypes can be calculated. Next session, Hardy-Weinberg law can be applied to human populations. So the first section is the CCR5 gene. The CCR5 gene, it is a gene that controls your susceptibility to HIV-1 infection. So the CCR5 gene, it encodes protein CCR5. It is a receptor for strains of HIV-1. So here is the CCR5 gene. 1, 2, 3, 4 introns. And then the last intron is delta 32. So some people are resistant to HIV-1. They are homozygous individuals resistant to HIV-1 infection. But people who are heterozygotes, they are susceptible but progress more slowly to AIDS. Genotype determined by direct DNA analysis using PCR and resident enzyme digest analysis. You get this genotype and phenotype. Genotype 1-1, susceptible to sexually transmitted strain of HIV-1. Genotype 1/delta 32, who are heterozygous, susceptible but may progress to AIDS slowly. But people who are homozygous for delta 32, they are resistant to most sexually transmitted strains of HIV-1. 1-1, that's the genotype. 32-32, that's the genotype. But 1-32 would have all three bands. So to compute frequencies, there are two methods. First of all is counting alleles, and next will be genotype frequencies. So let's say that we want to use the method counting alleles. So remember that each one of the humans would have 2 alleles per person. So if there is 79 people, how many alleles are there? How many alleles? So you times 2. 79 times 2 is 158. What if somebody is heterozygous, right? And then there are 20 of them. So if somebody is heterozygous, he only has one copy of one gene and the other copy for another. One copy for another. So if you have 20 people, right? So how many genes do you have? How many alleles do you have? So if you have 20 people, mean that you have 20 of the 1 allele and 20 of the delta 32 allele. And same goes with the 32-32, because this person is homozygous, just times 2. So if there is one person with this 32-32 genotype, mean that this person has 2 of that allele, 2 allele of 32-32. Next, what about from genotype frequencies? In this case, we are looking at who are the people. How many people are having the disease? How many people are not? And so, the person who have the disease, 79. Person who are having heterozygous, 20. Person who are resistant is 1. So how many people you have? 100. So from there, you can actually divide the number of people over the number of the total number of people. So what do you get? The frequency for 1-1, 79 over 100, 0.79. So for the heterozygous, 20 over 100, 0.20. So delta 32, 1 over 100, 0.01. Okay, so to look at the frequency of CCR5 in sample, how many do you have? Okay, you have 0.79, right? But for the heterozygous, you know that each one of the person will have only one allele in them. So just times 0.5 or half it, 0.20. So you get 0.89. What about the frequency of CCR5 delta 32? 0.5 times 0.20 because they're heterozygous, plus the people who are having the disease. How many? Okay. People who have the disease, it is 1 over 100, so it is 0.01. So you plus them both together, you get 11%. So recheck again, 89 plus 11, what do you get? You get 1.0. So how do we calculate frequencies of alleles? To calculate frequency of allele, you just capture the number of alleles over the number of total population. But what if you have multiple alleles? Okay, you add more additional variable. So let's say that there is 3 alleles controlling a trait, P, Q, and R. But one person can only have 2 alleles. So you add P square, Q square, R square plus PQ plus QR plus PR. Okay, so PQ, PR, QR are the heterozygotes. So P squared, Q squared, R squared, 2PQ, 2PR, 2QR, all totals to 1. For example, ABO blood type. ABO blood type, you have 3 types of blood. You have 3 types of alleles also, A, B, and O. But instead, one person can only have 2 alleles. So you are either AA, AO, BB, BO, or AB, or OO. And so how many alleles do we have? 3 alleles, but only 2 persons. So we need to also make sure that the AO, the BO, the AB are not left out. So if you can see here, frequency of allele IA is 0.38, allele for IB is 0.11, allele I is 0.51, and so P squared is what? It is the AA. AA is 0.38. So P squared is similar to, it is equals 0.38. So what do you get? You get 0.14. So that's the predicted frequency, 0.53. What about 2PR? Okay, first of all, before that, you have to find the R first. R is the O blood, or small I small i. Small I is 0.51. So if I find a point R, you have to square root it, you will get 0.26. Okay. And so from there you can find 2PR. So 2 times 0.38 times 0.51. Okay. Then Q 0.11 squared. So that's what you get. Q is BB, and 2QR is 2 times 0.11 times 0.51, you get 0.11. Then 2PQ is 2, 0.38, 0.11. What you get is 0.084. So it is quite a low number, because in real life, people with AB are quite rare to find. You combine all of the numbers, 0.53, 0.12, 0.08, 0.26. What do you get? You get 1.0. So that's when you know that your calculation is correct. Calculating frequencies of X-linked traits using Hardy-Weinberg equation. So for X-linked allele, do you remember? X-linked allele, only men will have the disease, woman will be carrier, and woman can also be normal. But for men, if a man is normal, means that he doesn't actually carry the allele at all. So let's say that a woman with an allele for both X chromosome will be Q squared. So heterozygote frequencies. Hardy-Weinberg law allows estimation of frequency of heterozygotes in the population. Frequency of recessive trait determined by counting individuals in population sample. And so first of all, you need to find the number of people who are recessive, and then you find the dominant. So you find R first, and then you find P, and then you find Q first, and then you find P, and then you can find the 2PQ. For example, in CF, there is an incidence of 1 over 2,500, which is 0.004 in the people of Northern European ancestry. And so, let's find the recessive allele first. The frequency is Q, square root it, you will get 0.004, you get 0.02. Okay. So the 0.02 is the P, the 0.02 is the Q, so you have to minus 1. 1 minus 0.02, you get 0.98. Okay, so 0.98 is what? Is P. To find 2PQ, 2 times 0.98 times 0.02, you get 0.04. 0.04 is also translated as 4%. Continued in part 2.